Okay, today we're working with a lab which is titled Cell Structure and Function. If you would please read through the introduction. It has some bold print terms which you will find necessary for the exam. And I'm going to begin with the section titled Prokaryotic versus Eukaryotic cells. Pro means before, karyon refers to the nucleus. These are cells that do not have a true nucleus, and that would be all of the bacteria. And then the eukaryotic cell would be all of the other cells besides the bacteria. And EU means true. Karyon refers to the kernel, which is what they first called the nucleus. And again, that's all of the other cells but the bacteria have a true nucleus, and so that would be plant, animal, fungus, and protist, so all of the others. So let's first look at the bacterium, which is the prokaryotic cell. If you'll notice here in the center, you see the DNA, and that looks like just a mass of threads, but that is really just one continuous circle of DNA, and there is no membrane around it. So bacteria only have one DNA, and it is a continuous loop, and it is not wrapped in a membrane. You do have some cytosol, which is the fluid that you find within within the cell, and of course you have a layer called the plasma membrane. Outside of that, all of your bacteria will have a cell wall that's helped maintain the structure of the bacterium. Outside of that, you may or may not have, this is called a glycocalyx, and the glycocalyx can be either fairly dense, and in that case it would be called a capsule, or it can be not very dense, and in that case it would be referred to as a slime layer. Okay, so some bacteria have these, some bacteria do not have these, and then on the exterior here you see these short thread-like looking structures. Those are called the fimbriae, and the fimbriae are structures that adhere to, say, like the mucous membrane, for example, of your oral cavity. If you happen to be exposed to one of those, then normally what happens if you have bacteria without the fimbriae, when you would swallow the saliva, the bacteria would go into your through your esophagus to your stomach, and then would be destroyed by the acid. Well, if you have the fimbriae, they tend to adhere to the walls of your mucous membrane, and therefore they do not go down with the saliva. And the longer they're on the mucous membrane attached, then the more copies of that they can produce. So they can divide about every 20 minutes. So it doesn't take long before you have massive numbers of these bacteria. So hence, because of that, you end up with a bacterial infection, which does happen. All right, then you see at the end of this we have a flagellum, and it is a whip-like structure used for motility. And as with the fimbriae, as with the glycocalyx, the capsule or slime layer, this structure is also not found on all bacteria. In fact, most bacteria do not have these, so it's not all that common, but some do, so they've included it just to give you an idea of what you might find on a bacterium. Okay, and so believe it or not, that is the simplest cell on the planet. So that one is the bacteria, which of course is the prokaryotic cell. Alright, so you have two things to do. One is to look at the slide of a bacterium, and it says a slide of a prokaryote. Well, that would be the bacterium. And you will find on that slide, you will find three different types of bacteria. The bacillus, which is a rod-shaped bacterium, the coccus, which is a spherical, kind of a baseball, Just what you will see will look like little dots. And then you have some spirilla, which of course would be spiraled shaped bacteria. Those are the three bacterial forms. And then it says examine a prepared slide of cuboidal cells from the human kidney. Well, I may not use that. I may put out a slide of an amoeba, which is has a huge nucleus, and you would have no trouble finding it. It's just easier to identify. So it would be could be any number of cells that I would use on that. Okay, and then when we get to the eukaryotic cells, now they're not including fungus here. They're only including the animal and the plant cells, and all of these cells will have a true nucleus. And again, eukaryotic means true nucleus in Latin. So they give you the structures that you'll find. Some of these structures are only found in plant cells. For example, the cell wall, the first one listed, the central vacuole, which I'm not finding it. Central vacuole. I'll show it to you anyway. And then the chloroplasts, which of course are used for photosynthesis. So that would also be found in plant cells. All right, and then plant cells include everything else that you have here on the list. Okay, now the animal cells, of course, are everything else that you have on the list. Now the one structure that you find in animal cells that you do not find in plant cells are what are called the centrioles, and I will point that out on the animal diagram that you will see. So they give you the name of the structure, which obviously you need to know the names of the structures. Also the functions of those structures, so you will need to know those as well. Okay, now going to the individual cells, this one is the animal cell, and you can see you have a nucleus, which is of course wrapped in it's a double membrane is what it really is, and it does have pores on it. And then within that you will find the chromosomes. Well, unless the cell is dividing, the chromosomes are in an uncoiled state, so they're not really visible, and at that point they're called the chromatin. And then also within that nucleus you see this round sphere that is called the nucleolus, which you'll find in your list above all of this. And the nucleolus is where the ribosomes are produced. The ribosomes are small enough to where they can actually get through these pores and go out into the cytosol. Some of them associate with the endoplasmic reticulum, which is a tubular system that you have outside the nucleus. And if the endoplasmic reticulum has the ribosomes attached to it, then it has a rough appearance. Not that you can see this under the microscope. It would take an electron microscope to see that. But so the ribosomes produce the proteins, and then they're transported through these tubes. And so this is called rough endoplasmic reticulum because the ribosomes attach. You'll also notice that you do have other tubes, and those tubes are called smooth endoplasmic reticulum, and that actually is where different medications can be detoxed. So it's a detoxification center. Also, it's a center where different types of lipids are produced. Fatty compounds, for example, phospholipids, which make up 45% of the plasma membrane, those will be produced there. Okay, further along here you see the mitochondrion, which is this structure. The mitochondrion is where all of the ATP is produced. And then next to that you see the Golgi bodies, or the Golgi apparatus. These are made of little flattened sacs. Looks like maybe a stack of pancakes perhaps. And if you'll notice that we have this budding that happens on the side of that, what really occurs in the Golgi apparatus is mostly a packaging. So you can actually send proteins into this and say lipids into this, and then package those together and then package it into a little membranous sac, which they call a vesicle, and that is what you're seeing being exported out of this cell. Sometimes these things that we're packaging together may be used within the cell. Sometimes they may be exported out of the cell. And so it's a repackaging center is really what the Golgi apparatus will do. Okay, and you see some more mitochondria. You see a peroxisome. Peroxisomes are used for detoxification of hydrogen peroxide. It's something that can damage a cell, so that's what that is used for. And then in the animal cell, you find a structure called the centrioles. Now these are not found in the plant cell, but they're found in the animal cell. And it's a barrel of tube-shaped structures, and there are actually two of these. I think you'll be able to see that when you look at this a little bit closer. Possibly, it's not too clear, but you have two barrel-shaped structures, one here and then one over here, of the tube-shaped structures. And the exact function of this is really not known, believe it or not. You would think by now they would have all the structures identified, which they have identified, but as far as the function of this is not really too clear. They do play a role in cell division, but like I said, plant cells do not have these. So that being the case, you would think a plant cell could not divide. It still divides. You can have one plant cell divide into two plant cells, but it doesn't have the centrioles. So they're certainly not absolutely necessary for cell division. So there again, they're not really too terribly sure about what that would be. Okay, now in the cytoplasm, you can see there are these tubes, little filaments that crisscross throughout the cytoplasm. There's some more like right in through here. What those do is to help maintain the shape of the cell, and then they also anchor organelles in place because of gravitational pull. In reality, everything that you have in the cell should actually be at the very bottom of the cell because of gravity, but because you've got things anchoring all of this into a very, very exact position, then things, of course, are not pulled by gravity to the bottom. And then, of course, on the animal cell, as on all cells, you do have a plasma membrane, and that does control what can enter and what can leave the cell. All right, moving on to the plant cell. There are structures that you find in the plant cell that you do not find in the animal cell. Initially, I think you will notice this big, huge sac on the interior of the plant cell. This is called a central vacuole. Primary function there would be the storage of water. It can store other things like pigments, waste products, but primarily water storage. The green structures that you're seeing that are kind of oval shaped, these are going to be chloroplasts, and that's where the plants produce their sugar by photosynthesis, so that's what that represents. And then the third thing that you'll find in a plant cell that you do not find in the animal cell is the cell wall. So here's the plasma membrane, the blue structure, and then outside that, here in green, would be the cell wall. Now the cell wall is really not a green structure, but they, I guess, colored it green just so that it's easily seen and distinguished from the plasma membrane. So it does give the cell some rigidity and some shape to the cell. It does make it difficult for things to get in and out of the cell, however, so plant cells have some difficulties that animal cells do not. Okay, so the one of the things that you'll work with would be a plant cell called an Elodea. Elodea is a water plant, and you'll be able to work with that. Look at that under the microscope. Just follow the directions. We're not changing anything about that section there. So if you can read, hopefully you can. And I lied. Actually, we are omitting number 9. It says save your slide. You don't need to save your slide on that. You're just making a wet mount of the Elodea. Okay. The other thing we're working with in this lab is how things get in and out of cells. One process is referred to as a diffusion. Diffusion is the movement of substance from an area of high concentration where you have more of it to a low concentration where you have less of it. So if you look at this dish, you can see that the crystal, which evidently this is some sort of a dye that they placed into this, I'm sure, well, they say in a semi-solid, but whatever that crystal is, it's a dye compound. And then, of course, outside, wherever they put the dye, then outside of that, then this area right here, you have a low concentration. In fact, you don't have any dye. So what's going to happen, the dye will move from where it's more concentrated into an area where you don't find any, or you find less of it. So it will move out in throughout. And on this, you're seeing it begin to move, and then at this point, you're seeing it equally distributed throughout that dish. So eventually, with a diffusion, you are moving something from an area of high concentration to low concentration, but eventually you do reach a state of equal distribution, which is, of course, what they would call an equilibrium. So that's what's happening with this. You're going to do that again through a semi-solid. It'll be a agar plate, and it kind of has a goo-like material in there, and you'll be able to see that, the semi-solid. We're also doing this through liquid, which will just be water. We'll use water and put a drop of dye, just like they did here, and then watch it diffuse through and calculate the amount of time it takes it to diffuse. And the other thing we're working with is diffusion through air. And so I will have some air freshener, and I will spray that, and then you will determine how long it is before it diffuses through the air before you can smell it. And we can calculate then diffusion rates through air, diffusion rates through water, and diffusion rates through a semi-solid, and determine those rates. Okay, then as far as this little experiment, what you're going to do on this, you've got a piece of what they refer to as a dialysis membrane, and at the bottom of it, it says closed in of dialysis membrane. I think they want you to close that with a rubber band. Typically, at this point, I would just tie that off. I would just physically tie it off and wouldn't actually put a rubber band around it. To me, that seems like that would leak. And then in this bag, what you're going to do, again reading the directions, you're going to put sugar, glucose, and iodine. Sugar and starch solution. Okay, and then out here in the beaker, you're going to place water and iodine. And so you're going to let it set for a period of time, and then we're going to see what things can move out of this bag and which of these substances can move into the bag. And we've got a little test we'll run on that. So just follow your directions. I don't believe there's anything we need to change on that. Just do what I've asked you to do on that one. Okay, on osmosis. Osmosis is a diffusion of water, which means water is going to move from where you have more water to where you have less water. That's just a simple diffusion, and they call it an osmosis. Now, on this experimental procedure, speed of osmosis, we're going to omit that. I've got an X beside that. So we're not doing this particular section here. We will pick up down below where you see tonicity, and let's discuss what that actually means. Tonicity is the concentration of the solute, which is whatever is dissolved. And in this case, it could be any number of things. It could be protein that's dissolved in water, could be salt, could be sugar. I think we'll use salt is probably the easiest thing to use. But if you see a term which they give you here, isotonic, iso is Latin for the same, and again tonicity refers to the concentration of whatever's dissolved, so concentration of solute. So this just tells us that the concentration of solute is the same both inside the cell and outside the cell. And they're using blood as an example. So if you put a 0.9% salt on a red blood cell, the red blood cell would not lose or gain water, because that is exactly the salt concentration of the red blood cell. Now, we're not doing that for a couple of reasons. One is we are certainly not going to work with blood. And the second reason is it's very difficult to get an exact 0.9 salt concentration. So that typically, even trying that, the results are not too good. Okay, notice another term that we have here, hypertonic. Now, hyper means more. Like a hyperactive child would be a more active child than, of course, what they should normally be. But in this case, we're looking at hypertonic. So that means more tonicity, which of course is more concentration of whatever is dissolved. So a higher concentration of the solution. So whatever solution we put onto the cell will be a higher concentration of solute, which is whatever is dissolved, than would be inside the cell. And we'll look at what happens there. The other term you see here is called hypotonic. Hypo means less than. So tonicity again refers to the concentration of whatever is dissolved. Salt, sugar, protein, whatever that would be. So this means there is less solute, sugar, salt, protein, in the solution than what you would have in the cell. And those, though, whether it's hypertonic or hypotonic, that affects the way that the water moves, and we'll look at that as far as how the water moves. So we're not doing the effective tonicity on red blood cells. You can omit that section. All right, now we are going to do the effective tonicity on the Elodea, which is the water plant. Okay, so what happens with that, and let's just go to a piece of paper here and just try to diagram this. Let's say that you have a plant cell, so it's somewhat boxy. Let's just use that. And most cells are going to be a 1% solute. Now that's a number of things: sugar, salt, protein, could be waste like ammonia, those things, which gives us a 99% water. Okay, outside that cell, let's place a 10% salt solution. It could be amino acid, it could be sugar, but I'm putting a 10% salt. That gives me a 90% water solution. Okay, so I have only a 1% solute in, 1% material dissolved inside the cell, and a 10% material dissolved outside the cell. So what this means is the exterior is hypertonic. Which essentially means more solute dissolved. And again, our solute is whatever gets dissolved. Well, that's referring to tonicity. Now let's go back to osmosis. Osmosis is the movement of water. And water is going to move from where you have more of it to where you have less of it. So if I have a 99% water inside the cell and a 90% water outside the cell, obviously water will leave that cell. So what's going to happen once we put that cell in a 10% salt solution, the plasma membrane is going to shrivel up and shrink up and pull away from the cell wall, and you should be able to notice that. The term given for that is called plasmolysis, or plasmolysis, whichever. And if you bust the word apart, look at it, lysis means to break, and the plasma refers to the plasma membrane. So this means that the plasma membrane was broken. Well, it wasn't broken at all, it actually just shriveled up. But, so this is what we would call a misnomer. It's not a good term for what's happening, but nonetheless, they still use it, and maybe someday they'll change it to something appropriate, but for now we still use plasmolysis. So the plasma membrane pulls away from the cell wall, and it's kind of like letting water out of a water balloon. It just shrivels up. Now let's go to the other situation, which would be a hypotonic solution. So once again, cells are about a 1% solute, sugars, salts, amino acids, waste products. So that's a 99% water there. And then outside this cell, let's put 100% water. Okay, so if it's 100%, we don't have anything dissolved in it, so that's 0% solute. Nothing is dissolved in that. So what happens then, this is obviously a hypotonic solution. So the amount of solute is less outside than it is inside. 0% solute is less than 1%. So hypotonic, less solute. This solution has less solute. In fact, it has no solute. So it must be less than if you have any solute inside the cell. Okay, so that's tonicity. The other thing you need to consider here is the movement of water, which is osmosis. One more time, water is going to move from where you have more water to where you have less water. So in this case, the water is going to move in, and that does create a tremendous amount of pressure inside this cell. The cell will not break, it will not burst, because you do have a rigid cell wall on it. So it creates a pressure. They call that a Turgor pressure. Again, that's the term you would use for plants. With animals, it's a little bit different. So the water moves in. You do not break the plasma membrane or the cell wall. It just creates a pressure because of the osmosis, the water moving from the outside to the interior. It just creates pressure there. Okay, now on an animal cell, this is going to be different. The terms are different. So we're using an animal cell now. One more time, this is a 1% solute. That's pretty common for most cells. There are some that it would be a little different on, but we're looking at your common cell. Okay, so 1% solute, that gives you a 99% water. Outside our solution that we'll put this Elodea in, well, in this case, in just an animal cell, any animal cell, then we'd use say another 10% salt. So that's my solute, is the salt. Could have been sugar, but we're using the salt. That only leaves me with a 90% water. Okay, now outside you have more solute, the salt, whatever's dissolved, than you do inside. So this cell is placed in a hypertonic solution. The tonicity is greater, the concentration of solute is greater outside, the 10%, than it is inside, which is the 1%. So the solution outside is hypertonic. Now also, once you've done that, then you have to consider the concentration of water, which is osmosis. Water moves from where you have more of it to where you have less of it. So, of course, what's going to happen, this cell is literally going to shrink up. And that's what we did with the plant. But we called it a plasmolysis. In animals, we call this a crenation. So the term does change. The mechanism is the same, just the movement of the water moving out of the cell. And so it just shrivels up, and that is a crenation. Now if we had another animal cell, let's put in our 1% solute, and that is again a 99% water. Outside, let's put 0% solute, so we don't have anything dissolved. That means we must be working with distilled water, 100% water is what we have. Okay, now The outside has less solute. It has no solute, which has to be less than 1%. So the outside of this cell is going to be hypotonic. It has less solute. Tonicity means the concentration of the solute. So the outside has 0, inside has 1. So outside has less stuff dissolved, which is hypotonic. Okay, then consider your water. Water moves from where you have more of it to where you have less of it. Well, I've got 100% outside the cell, only 99% inside the cell. So in this case, the water is going to move to the interior of the cell. Well, because we don't have a rigid cell wall around this animal cell, it will break. It will just burst. It's like putting too much water in a water balloon. That is called a lysis, which means to break. So if you fill a water balloon up with too much water, which is basically what we're doing here, except it's an animal cell, we're going from a high concentration of water, 100%, to 99%, yes, water will move in. It's called osmosis. An animal cell can only hold so much water, so of course that is going to break, which is a lysis. All right. Now, the other thing that you're working with here is the potato strip procedure. There isn't anything that we need to change on that. Just follow that as written. And we will stop at that page. So once you're finished with the potato strip procedure, then you will be finished with the lab. Okay, well, that's another lab. And thank you. Thank you for your time.